NINHYDRIN is a chemical that reacts specifically with this amino acid. When the reaction happens, it produces a deep purple compound known as Bohemian's purple. When we touch a surface, we leave behind tiny amounts of sweat, mainly from our S-ring glands, which contain amino acids. And this amino acid will form a pattern that matches the ridges of our fingerprint. If someone has recently touched their face or scalp, secretion from sebaceous gland may also be transferred. And this purple color develops exactly where the amino acids are deposited. So, the fingerprint becomes visible to the naked eye. Interestingly, the intensity and shade of the purple can vary depending on the type of amino acid present. This technique works particularly well on porous materials like paper, which absorb the sweat and amino acids better than non-porous surfaces. That makes NINHYDRIN an ideal method for document-based fingerprint detection. The goal is to create a method that can both detect the fingerprint, the latent fingerprint, and drugs at the same time. Many illicit substances such as cocaine and methamphetamine, it fall under the amine classification, and this can be visualized by using NINHYDRIN, which will react with primary and secondary amine. And this type of amino acid which can be found in sweats alongside with certain drugs. So if drugs are present on the surface, they may become visible alongside the fingerprint, allowing for presumptive drug detection during the fingerprint development. And this approach can help reduce the analysis time since only one test is needed instead of several. It can also flag the presence of drug if they were not originally suspected, making it easier to decide which samples need further testing. Overall, this dual-purpose method can help make the forensic investigation faster and more efficient.
From this research, there is one question that arises. Can NINHYDRIN fingerprint analysis on drug-infused papers be used as a presumptive method to indicate the presence and possibly the class of certain illicit drugs by observing variations in the color and intensity of developed fingerprints compared to drug-free controls? This question arises from the observation that NINHYDRIN, traditionally used to detect latent fingerprints on paper, produces different colors when reacting with fingerprints on papers infused with certain drugs, such as amphetamines, opioids, and cocaine, suggesting a potential dual-purpose forensic tool for both fingerprint and presumptive drug detection. The objectives of this approach focus on creating a more efficient and effective way to detect important information on paper materials. One goal is to combine two common detection tasks, finding fingerprints and identifying certain substances, into a single streamlined process. This simplifies the work by using one method to reveal both types of evidence. Another aim is to enhance the ability to spot attempts to hide substances within paper items, such as letters or cards. This is particularly useful in environments where security is critical and quick identification is necessary. Additionally, this method helps reduce the workload and time needed for analysis. By allowing both types of evidence to be checked at once, it minimizes the need for multiple separate tests, making the overall process faster and more cost-effective. Finally, the approach supports quick, on-site preliminary testing. This means that materials can be rapidly screened visually, so only those that show signs of containing certain substances are sent for more detailed examination. Overall, this method offers a practical solution that improves detection capabilities, saves time and resources, and supports faster decision-making in various security and investigative settings.
The drugs used in this research ranges from common drugs to synthetic cannabinoids. Among the drugs that are used in this research are cocaine, and methamphetamine, MDMA, and synthetic cannabinoids like AKB48 and THJ018. The chemicals used are obviously NINHYDRIN, which is the subject of our research, absolute ethanol, ethyl acetate, and the Novak 71DE engineering fluid will be mixed along with the NINHYDRIN to create a working solution, while the methanol and acetylnitrile is the medium used to prepare the drugs. And lastly, the other materials that are essential to the research, which includes the fingerprints on the paper samples from our donors, and surface material like A4 paper, magazine paper, and greeting card. So now let's look at the methodology of this research. The process of this experiment starts with the infusion of drug into paper. Drugs are prepared at 50 mg per milliliter in methanol, except for the two synthetic cannabinoids, which will be prepared in acetylnitrile instead. So the drugs will then be dropped at the center of each paper, cut into a 3 cm by 3 cm square, with each sample being produced in a triplicate, which means the total of sample each drug should have is 9. Blank samples were also prepared for each 3 paper type. One is a sample that is infused, a paper that is infused with pure methanol. The second one is infused with pure acetylnitrile, and the last one is not infused with anything, which just means it's just a blank paper. Next, we will look at how the NINHYDRIN is introduced to the sample. Before addition, the NINHYDRIN must first be made into a working solution. the ninhydrin is mixed with absolute ethanol, ethyl acetate, followed by acetic acid, and then it will form a slurry, and then Novec 71DE engineering fluid will complete the mixture. So the solution is put aside first, as we will now look at the infused papers. 1 to 2 fingerprints were deposited on the surface of the paper by a single donor, which means a single paper will have fingerprints from the same donor. Although actually in this research we have 2 persons as the donor. So not all papers would have fingerprints of the same donor, which means for F4 paper with morphine, we'll have our donor A, and for cocaine, we'll have donor B. So all 10 fingers of the donors must be used to be deposited on the paper, and some will be primed, and while others are non-primed. Well, the prime in this concept, in this research, is actually the fingerprints that are primed are actually touched on the forehead first, and the non-primed is just pure fingerprints. Well, then the sample papers are then fully soaked into the ninhydrin solution made earlier. So the solution must be changed for every different type of drugs infused. The soaked samples is then left in the humidity chamber at 70 degrees Celsius for 15 minutes, and after 15 minutes have passed, the samples are then removed and observations are recorded.
So moving to the next part, which is the result. So the focus of the study is to investigate the use of ninhydrin for latent fingerprint on drug-infused paper. So in this research, they done the control sample in order to set a benchmark or reference to ensure the accuracy. So from the table, as we can see, there are 3 types of sample: blank, methanol, and also acetylene blank. And also all the color of the fingerprint turns out purple, pink, purple, and also purple. So this shows that without the presence of drug, there is no color change of the fingerprint. So from the picture, as we can see, there is the normal fingerprint detection using ninhydrin. So the next table shows the description of the fingerprint colors over the drug-infused paper on the 3 types of paper, which is A4, magazine, and also greeting card. So from the figure also, we can observe that amphetamine, it has the whole stained dark area purple on the fingerprint. Cocaine, it turns out grey and blue, and also morphine, it turns out light and grey on the fingerprint. So this indicates that there is a presence of the drugs on the fingerprint. However, no color changes occur for barbiturates and also synthetic cannabinoids. Furthermore, it was found that a visible fingerprint could be obtained on the drug-infused paper and also uninfused paper. So a significant observation was that the paper that infused with amine types of drug consistently show variation fingerprint color between the infused and also the uninfused paper, where it actually provide a clear indication the presence of the drugs. So from the table 1, blank paper and solvent blank samples consistently produce a purple fingerprint, which is actually the expected result for the ninhydrin. So as we can see, there were no color variation occur across the full fingerprint between the different types of paper in the blank sample. So moving to the next table, what we can observe was there are specific color variation were noted on the fingerprint that has the drug infused area. So what we can see was amphetamine class drug and morphine resulted in the dark purple color, meanwhile dimorphine and cocaine appear in the light grey or blue. So actually the color altered can be observed on the portion of the fingerprint which has the drug infused. So amphetamine sulfate was unique. So the entire dark, the entire paper that has the fingerprint turns dark stain because this was attributed to the primary amine tested, suggesting that the primary reaction between the ninhydrin and the drug itself. So meanwhile, barbiturate and synthetic cannabinoid did not cause any color variation on the fingerprint that has the drug infused area. So this is because amides cannot react with the ninhydrin. So next, the study highlighted that ninhydrin is very useful in detecting primary and secondary amine and amino acid. So the observed color variation for the secondary and tertiary amine like methamphetamine, MDMA, cocaine, and morphine suggested that ninhydrin reaction in this case primarily involved the amino acid from the fingerprint residue, with the drug affecting the visual outcome. So next, the advantages and limitations for this test. So the advantages are it can detect simultaneous detection of fingerprint and drugs. So in one sample, we can detect the fingerprint and also we can detect the drugs. So it is actually the cost-effective and time-saving test. Moreover, it is useful for initial screening, where it is actually the presumptive test where we can done on the crime scene. So the limitation is it is not a confirmatory test. So to confirm the types of drug, we need to do a further test. And then it is also detect the limited drug class detection. So as my explanation before, it can be only the NINHYDRIN can be only detect the primary amine. So the amide cannot be detected by NINHYDRIN. So this has a potential for false negative result for certain drugs.
There are a few discoveries made from this research. NINHYDRIN, a chemical commonly used to reveal fingerprints on paper, can do more than just show prints. When applied to paper that contains certain drugs, it causes visible color changes in the fingerprints, which are different from those on clean paper. The colors vary depending on the type of drug present. For example, papers with some drugs like amphetamine show a dark purple stain across the whole area. Other drugs, such as methamphetamine, cocaine, or morphine, cause color changes only in the fingerprint area, showing shades of purple, blue, or gray. However, some drugs like barbiturates and synthetic cannabinoids do not cause any color change, so fingerprints look the same as on drug-free paper. This discovery means that NINHYDRIN can be used as a quick visual test to suggest the presence and type of certain drugs on paper. It acts as a dual-purpose tool, helping to find fingerprints while also indicating if some illicit drugs might be present. Using this method can make forensic and security work more efficient. For example, it allows quick screening of paper items such as prison mail for both fingerprints and drugs at the same time. This reduces the need for multiple tests and helps focus detailed analysis only on suspicious samples. It's important to note that this method only works for drugs with certain chemical groups. Drugs without these groups won't show any color changes. Still, this research opens the door for further improvements and new ways to detect drugs on paper more easily and accurately. In summary, NINHYDRIN fingerprint analysis on drug-infused paper can act as a rapid, presumptive indicator for the presence of certain illicit drugs, particularly those with amine groups, while simultaneously revealing latent fingerprints. This dual functionality can improve the speed and efficiency of forensic investigations.